The overall goal of this procedure is to induce and visualize cyst wall formation in Toxoplasma gondii in vitro. This is accomplished by first culturing macrophages and fibroblasts, and then by infecting these host cells with the parasite. Stress responses can be induced in T. gondii by shocking fibroblasts with pH or activating the macrophages, which will both lead to cyst wall formation. Finally, fluorescent lectin is added to the cultures, which binds to the cyst wall, such that cysts can be visualized with immunofluorescence microscopy.
Hi, I'm Crystal Tobin from the laboratory of Dr. Laura Knoll in the Department of Medical Microbiology and Immunology at the University of Wisconsin-Madison.
I'm Angela Pollard, also from the Knoll lab.
Today we will show you a procedure for Toxoplasma gondii cyst wall formation in vitro.
We use this procedure in our laboratory to study parasite responses to stress conditions.
So let's get started.
Begin by placing a sterile round glass cover slip into the bottom of each well of a 24-well tissue culture plate. To induce the conversion of Toxoplasma gondii to bradyzoites in human foreskin fibroblasts under conditions of high pH and carbon dioxide starvation, start with a confluent 150 square centimeter flask of human foreskin fibroblasts. Under a tissue culture hood, rinse the confluent flask of human foreskin fibroblasts 2 times with 1x PBS and then add 2.5 milliliters of 0.025% trypsin EDTA. Incubate the flask at 37 degrees Celsius for 5 to 10 minutes.